Welcome, everyone. Today, we are going to embark on a fascinating journey back in time to explore the theories of chordate origin. If you have ever wondered where we, as vertebrates, truly come from, you are in the right place. We will uncover how scientists trace our biological family tree across millions of years, looking past superficial appearances to find our true invertebrate ancestors. To begin our exploration, imagine you are a detective trying to trace your family tree back millions of years. Now that we have set the stage, think about the challenge here. All the birth certificates and paper records of evolution have been completely destroyed. Instead of written documents, evolutionary biologists only have scattered clues. They look at the shape of a distant relative's face, the way they walk, or how they develop as a baby. Nature is full of surprises, and as we will see, the true story of our origins involves microscopic marine larvae and sea stars, rather than the creatures we might initially suspect. Before we dive into the specific theories, let us clearly define who we are talking about. Chordates are a major group of animals that includes a vast variety of creatures you know well, such as fishes, birds, dogs, and even humans. Despite our obvious differences in adult form, all chordates share a common biological blueprint. The most famous feature of this blueprint is the presence of a spinal cord, or nerve cord. For a long time, scientists looked at other animals with long bodies, wondering if our ancient ancestors looked like them. This fundamental question drove the earliest theories of our origins. This brings us to the first major attempts to solve the mystery. Over the past century, scientists proposed several models, notably the annelid theory and the arthropod theory. These early hypotheses suggested that chordates evolved from segmented worms, or insects. To make this work anatomically, scientists proposed that these ancient creatures flipped their bodies completely upside down. This rather extreme idea was an early attempt to explain a key chordate feature: why our nerve cord is located on our back, or dorsal side, rather than on our belly like a worm. However, as biological techniques improved, these theories began to show significant flaws. Moving past the idea of upside-down worms, we arrive at the most widely accepted and scientifically supported model today, the echinoderm theory. This theory is specifically centered around the diplerula concept. It posits that chordates and echinoderms, which include animals like starfish and sea urchins, actually share a very recent common ancestor. Instead of looking at adult bodies, biologists found striking similarities in their embryonic development and larval stages. By studying these early developmental pathways, scientists can formally define the evolutionary bridge that connects simple marine invertebrates to complex vertebrates. It is a beautiful example of how early life stages reveal our deepest history. Now that we understand what the echinoderm theory is, you might wonder why it matters what a microscopic larva looked like 500 million years ago. Understanding the origin of chordates is absolutely crucial because it explains the fundamental architectural blueprint of our own human bodies. It tells us exactly why our nervous system develops on our back, how our internal organs are arranged, and why we even possess a tail during our early embryonic stages. In the real world, this foundational knowledge is heavily used in advanced medical research. When scientists study genetic diseases or developmental birth defects, they rely heavily on these ancient evolutionary pathways. To move beyond just observing these traits, scientists need a way to quantify them. In evolutionary biology, scientists do not just guess which theory is correct. They use mathematical models. To evaluate whether chordates came from annelids or echinoderms, biologists use a metric called the simple matching coefficient. The formula is S equals M divided by the sum of M and U. Here, S represents the similarity index, which is always a decimal value between 0 and 1. The variable M stands for the number of matching traits that both groups share. The variable U represents the number of unique or unmatched traits belonging to only one group. When matching traits increase, the index gets closer to 1, indicating a very close evolutionary relationship. Now that we have our formula, let us test the annelid theory of chordate origin. Suppose a biologist examines a total of 20 fundamental anatomical traits between an earthworm and a primitive chordate. They find that only 4 traits match, such as bilateral symmetry, so M equals 4. Meanwhile, 16 traits are completely unique, so U equals 16. We substitute these exact values into our formula. In the denominator, we add 4 and 16 to get 20, representing the total distinct traits analyzed. Dividing 4 by 20 gives us our final answer of 0.20. This low score mathematically demonstrates a very weak relationship. So we have calculated a similarity index of 0.20. Let us prove what this actually means for the theory. This decimal translates to a mere 20% overlap in traits. This mathematically proves that annelids and chordates share very few fundamental developmental characteristics. Because this value is so incredibly low, biologists concluded that the annelid theory is highly unlikely to be true. The math simply does not support the anatomy. This realization prompted the scientific community to abandon the worm hypothesis entirely and search for a much better evolutionary ancestor, leading us directly to the echinoderms. Having rejected the annelid theory, let us apply the exact same mathematical logic to the widely accepted echinoderm theory. A biologist compares the embryonic development of a starfish larva to a primitive chordate. Out of 25 fundamental traits examined, they find that 21 traits match perfectly, so M equals 21. Only 4 traits are unique, meaning U equals 4. We plug these numbers into our equation. We add 21 and 4 in the denominator to get 25 total traits. Dividing 21 by 25 yields a final similarity index of 0.84. This is a remarkably high score. Now that we have calculated this new index, let us look at the proof it provides. A similarity index of 0.84 means there is an 84% match between the two groups. This is a remarkably high score for 2 seemingly different groups of animals, like a starfish and a primitive fish. This high mathematical correlation strongly supports the echinoderm theory as the most accurate model. It proves that chordates and echinoderms share a very recent common ancestor, which most likely resembled the hypothetical Diplurula larva. The math perfectly aligns with the biological evidence. To visualize what we have just proven mathematically, let us look at this phylogenetic tree. At the very bottom, you see the root of our tree, labeled as the hypothetical Diplurula ancestor. From this single ancient starting point, the lineage splits into 2 main branches. On the left, we have the Echinodermata, represented by the starfish. On the right, we have our own phylum, the Chordata. Notice the specific evolutionary milestones marked along the chordate branch. These include the development of the notochord, the dorsal hollow nerve cord, and pharyngeal slits. This diagram beautifully summarizes how a simple marine larva eventually gave rise to complex vertebrates. We have covered a lot of ground today, so let us summarize the key takeaways. First, we learned that the origin of chordates was a major puzzle, solved by focusing deeply on embryonic development rather than superficial adult appearances. Second, while early theories tried to link us to worms and insects, mathematical tools like the simple matching coefficient proved those relationships were incredibly weak. Finally, the biological and mathematical evidence overwhelmingly points to the echinoderm theory. By sharing a common ancestor similar to the Diplorula larva, echinoderms and chordates are proven to be evolutionary cousins. Great job following along with this incredible journey of life on Earth.